At the turn of the millennium, genetic sequencing shifted from mapping single strands of DNA to processing biological data through high-capacity pipelines. In 2003, 454 Life Sciences launched the first commercially viable next-generation sequencing platform, ending the long-standing monopoly of the traditional Sanger method. By 2007, Roche acquired the company, positioning the 454 architecture as the definitive standard for the future of genomics. Yet, despite this early market lead, the entire platform was discontinued by 2013. This horizontal bar chart compares average read lengths across these early platforms. Read length measures the physical number of consecutive base pairs decoded from a single continuous DNA fragment. As the data indicates, 454 held a substantial competitive advantage over early Illumina machines. Producing reads of up to 600 base pairs provided crucial genomic context, making it much easier to assemble and map genomes. The second critical metric is sequencing depth, or coverage. This is a statistical measure of how many times a specific nucleotide is redundantly sequenced across all the reads in a single run. While traditional Sanger sequencing processed one read per reaction, 454 increased sequencing depth by scaling up to millions of parallel reactions running simultaneously. 454 combined long sequence reads with high parallel throughput. To understand why a technology with these specifications failed, we have to reverse engineer its architecture down to the atomic level. The 454 workflow begins with library preparation. Genomic DNA is physically fractured into manageable segments, specifically targeting fragments roughly 300 to 700 base pairs long. Through a process called adapter ligation, synthetic sequences are attached to the ends of these fragments, including a unique 8 to 10 base pair barcode on the 3' end. This barcode enables sample multiplexing. It allows researchers to mix up to 96 distinct patient samples into a single sequencing run, which immediately reduces the cost per sample. But this creates a core engineering challenge. If you mix millions of distinct DNA fragments in a bulk fluid, amplifying their signals will cause them to blend into an unreadable noise. The mechanical solution is emulsion PCR. The fragmented DNA is violently mixed with a dense oil, forcing the liquid to shatter into uniform, isolated microdroplets. Inside each microreactor is exactly one microscopic bead, one single DNA fragment, and the necessary PCR reagents. Amplification takes place entirely inside the droplet. The DNA fragment is continuously copied until 1 million identical clones are anchored to that single bead. Emulsion PCR overcomes the parallelization bottleneck. It transforms a bulk mixture into 1 million perfectly isolated signal amplifiers, preventing cross-contamination before the reading process begins. Once the beads are amplified, the process moves to the physical hardware. This relies on the pico-titer plate, a grid where each individual well holds a volume of exactly 10 to the negative 12th liters. The emulsion is broken, and the millions of amplified beads are physically deposited across the surface of the plate. The geometry of the plate is specific. Each well is sized to fit one single bead, along with the precise set of sequencing enzymes required to drive the reaction. Because the wells are so small, hundreds of thousands of these individual reactions occur simultaneously in the same physical space. Instead of running a single bulk reaction to read one strand of DNA, the machine reads the entire plate at once. The pico-titer plate functions as a parallel biological hard drive, locking hundreds of thousands of distinct genetic codes in place so they can be read at the exact same moment. The readout relies on a process called pyrosequencing. In each cycle, the machine washes the entire plate with a fluid containing only one of the 4 types of nucleotides at a time. If the DNA template requires the nucleotide washing over it, polymerase incorporates the base, physically cleaving off pyrophosphate. Pyrophosphate reacts with APS and ATP sulfurylase to synthesize ATP. ATP interacts with luciferin and oxygen, producing oxyluciferin and emitting a single photon of light. An optical photomultiplier tube captures this flash. Because the machine identifies which nucleotide fluid was used in that cycle, a flash of light identifies the base pair. Pyrosequencing translates the microscopic addition of a single molecule into a macroscopic flash of light captured by a digital sensor. But hidden within this biochemical cascade is a physics limitation, a problem triggered entirely by homopolymers. A homopolymer is a sequence of identical bases occurring consecutively in a genome, such as 7 A's in a row. Because the machine washes the plate one nucleotide type at a time, Encountering a homopolymer means the polymerase incorporates all the identical bases simultaneously in a single cycle. This line graph maps the number of bases incorporated against the intensity of the light emitted. The math is direct. Incorporating one base yields one photon. Incorporating 3 bases yields 3 photons. At this scale, the light output is predictable and linear. But as the homopolymer stretches to 6, 7, or 8 identical bases, the physical scaling breaks down. The signal flattens out, producing roughly 4 to 5 photons, even when 8 bases are attached. At this intensity, the optical sensor cannot distinguish the brightness of 6 A's from 7 or 8. The error margins overlap completely. The data consequence is immediate. If the true genetic sequence has 8 A's, the sensor registers the muted light output and records only 7. This is an inescapable physics limitation of photon emission and detection. It guarantees insertion and deletion errors whenever the machine tries to read repetitive DNA. In genomics, an insertion or deletion error destroys data integrity. Dropping a single base causes a reading frame shift, meaning every subsequent base is parsed incorrectly and outputs non-functional protein data. This limitation rendered 454 technology largely unusable for microbial genomics and complex eukaryotes, which are filled with low-complexity regions and tandem repeats. The final economic pressure came from Illumina. By 2010, the 454 system was expensive to operate. Using specialized enzymes and exhaustive wash cycles pushed the cost to nearly $1,000 per million reads. Meanwhile, Illumina's cost efficiency was improving rapidly, dropping the price for the same data volume toward $50. 454 offered superior read lengths, but it could not justify a premium price while producing systematic errors on standard homopolymers. 454 Life Sciences initiated the next-generation sequencing era through brilliant engineering, but the platform ultimately serves as a lesson in bioengineering. A system is only as viable as the physical limits of its readout mechanism.